Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuhu. Hope so students you all are good, fine and healthy. Well, in our previous lecture we have discussed about the mitosis as well as we have discussed about the mitosis phases and the cytokinesis that is going to occur after the mitosis. Now in this video we are going to learn about the meiosis prophase 1. So let's see what is basically meiosis. Basically, students, meiosis comes from the Greek word. It is basically a Greek word, means meon. Meon means to get smaller, to reduce. Now, what actually it means? Basically, in meiosis, a one parental cell having the 46 chromosomes used to produce the 4 daughter cells having the 23 number of the chromosomes. This means that the chromosome number used to get reduced in the daughter cell. So that's why it is named as the meiosis. The definition of meiosis is that a parental cell is going to produce the 4 daughter cell having the half number of the chromosomes of the parental cell. In the parental cell, basically the meiosis, the parental cell in the meiosis is basically diploid and it used to produce the 4 haploid daughter cell. Now, what is basically haploid or diploid? Basically, diploid means 2n and haploid means 1n. The 2n means the 46 number of the chromosomes while 1n means 23 number of the chromosomes. If you're going to discuss about the cell cycle of the meiosis, then it will start from the interphase. The interphase is totally similar to the mitotic interphase. It will include G1 phase followed by S phase and then the G2 phase. After that, the meiosis 1 used to occur. And after meiosis 1, there is a meiosis 2. In meiosis 1, the karyokinesis and the cytokinesis used to occur. And cytokinesis. The karyokinesis will have the 4 phases like prophase 1, metaphase 1, anaphase 1 and telophase 1. Basically in meiosis 1, the parental cell having the diploid number of the chromosomes will produce 2 haploid number of the chromosomes, daughter cell. In meiosis 1, the diploid mother cell is going to produce the 2 haploid daughter cells. Basically, the meiosis 1 is the step in which the genetic variation used to occur. What is basically the genetic variation? The variation means change and genetics means in gene. So, the change in the gene will be known as the genetic variation and the best example is you. You might have seen that your brother and sisters while production of the same parents but are changed from each other. Some is more intelligent and some is more beautiful and someone is more heighted and others are smaller. So how this come while you all are basically the production of the same parents? So how this genetic variation used to occur in you? Yes, this is basically due to the meiosis 1. Now we are going to learn that how this genetic variation used to occur in the meiosis 1. Let's learn about the meiosis 1. The first and the most important phase of the meiosis 1 is the prophase 1. Prophase 1 is the phase in which the genetic variation used to occur. Basically, in the Prophase 1, the thread-like structure of the chromatin used to condense and form a chromosome. These chromosomes used to align with each other. The homologous chromosomes will align with each other. Now, what is the meaning of homologous? The homologous are the chromosomes which are similar to each other. So, this one chromosome is similar to this other chromosome. So, the similar chromosomes are used to align with each other. And this alignment will be known as the synapses. Basically, the synapses is the procedure in which the two homologous chromosomes are going to align with each other, like standing besides of each other. After the synapses, basically this is the chromosomes having a sister chromatin. It is going to align with the chromosome that is a homologous of each other. After the synapses, the next step is a chiasmata. In the chiasmata, the non-sister chromatins are going to join with each other at some specific points. Let's suppose this is a chromosome. And this is also another chromosome, and their sister chromatins are going to join with each other at some specific points. This is basically known as a chiasmata. This is basically one chromosome and this is another chromosome. The non-sister chromatin basically used to join at some specific points with each other, and this joining with each other will be known as a chiasmata. These are two sister chromatin and these are two sister chromatin, and these basically are non-sister chromatin. After the formation of the chiasmata, the chromosomes are going to exchange their genetic information. This is like exchange. These both are going to exchange their genetic information, and exchanging of the genetic information will be known as a crossing over. And this exchange of material will result in the genetic variation. Let me explain you in very much detail that what is happening. Let's suppose these two are the homologous chromosomes. This is one chromosome and this is another chromosomes. They both are the homologous to each other, so they used to align with each other, and this is known as a synapses. After the synapses, the chiasmata will occur. In chiasmata, the non-sister chromatin are going to join with each other at some specific points. And this joining of the non-sister chromatin with each other is known as a chiasmata. After the formation of the chiasmata, as I told you before, the exchange of the genetic material will occur. And this exchange of the genetic material is known as the crossing over. Now, how the crossing over is going to result? Let's suppose this is one chromosome. After the crossing over, it will appear like that. Well, the other chromosome is going to appear like that. So basically, this was the point of the joining of the chromosomes, non-sister chromatin with each other, and now they have exchanged a genetic material with each other. After the crossing over, the chromosomes are going to have a new leg. Now you can see in this diagram, these are basically two homologous chromosomes which are going to align with each other, and this is known as a synapses. Now these are basically the points in which they are basically joining with each other along their length. As this joining of the point along their length will be known as a chiasmata. After that, they are going to exchange their material. The non-chromatin sisters are going to exchange their material with each other. After the exchanging the material, crossing over, these chromosomes are going to appear like that. This chromosome have shared this part from its non-sister chromatin chromosome, and this chromosome have shared this part from its non-sister chromatin chromosome. Now you can see that these chromosomes are basically different from their original chromosomes. This is known as a crossing over, and this basically, students, this crossing over will bring the genetic variation. That's why we are actually different from each other. After genetic variation, now what will happen in the prophase after the genetic variation? The chromosomes will remain bivalent. As means the chromosomes will remain like that. Tetrate, you can say. The chromosome will remain in that condition, and they are going to get more condensed. They are going to further condense themselves, as well as in the cell, the nucleus membrane will disintegrate or will be removed. Similarly, the nucleolus present in the nucleus will also degenerate, and this will be already learned in the mitosis. The nuclear membrane will disintegrate, degenerate. Similarly, the nucleolus which is present in the nucleus will also disappear. Now what will happen? The bivalent chromosomes will separate themselves in the nucleus. Moreover, the centrioles, which are basically duplicate in the interphase, are going to move toward the opposite pole of each other, and they are going to produce the spindle fiber. And you know it very well, these spindle fibers which are going to attach with the kinetochore protein will be known as a kinetochore spindle fiber. And you know very well that each bivalent chromosomes is going to attach from each pole, that each bivalent chromosomes is going to attach from both kinetochore spindle fiber, from two kinetochore spindle fiber from each pole. Similarly, these kinetochore fibers will produce and will join with the kinetochore protein of the chromosomes from the both pole, as well as the non-kinetochore fiber will also produce, which are going to join with each other. This we have already learned. So this is basically the completion of the prophase 1. Now student, the next step is the metaphase. In the metaphase 1, these actually bivalent chromosomes are going to arrange themselves in the center of the cell and will form the metaphase plate. So in the cell, these bivalent chromosomes are going to arrange themselves in the center of the cell. And this non-kinetochore and kinetochore spindle fiber will separate and will attach with the kinetochore protein from both centrioles. In the Metaphase 1, the Metaphase is going to make a Metaphase Plate. So in Metaphase 1, you have seen that the bivalent chromosomes are going to arrange themselves in the equator and will form the Metaphase Plate. After the formation of the Metaphase Plate, the next step comes Anaphase. Anaphase 1. In the Anaphase 1, what will happen? Each kinetochore spindle fiber will become shorter and they are going to pull one chromosome toward each other. When this kinetochore will become shorter and shorter, they are going to pull the chromosomes toward each other. And the non-kinetochore fiber will start elongated themselves, and they are going to appear just like that. So, the non-kinetochore fibers will start elongated themselves. This is one centriole and this is the other centrosome. Non-kinetochore will elongate themselves while the kinetochore fiber will start shortening themselves. And they are going to pull the chromosomes toward each other, toward themselves. The kinetochore fiber will become shorter and will start pulling one chromosome from a bivalent chromosome. They are going to pull one chromosome toward itself, and this is known as the anaphase. After the anaphase, the next phase is the telophase. The telophase 1, you know very well, in the telophase 1, the cell will start forming the furor, the cleavage furor, as well as you know very well that the telophase is the inverse of the prophase. Now, what is going to happen, student? Here, basically, this spindle fiber will start removing their cell. The chromosome membrane, the nuclear membrane, is going to reappear again. The centrioles will present in each cell. You can say one centrosome in each cell. And one centrosome, you know it very well, I have told you before, the one centrosome means, yes, two centrioles. As well as students, one more thing is going to happen. The nuclear membrane will start forming, as well as the nucleolus will also appear. You know very well that the telophase is basically the reverse of the prophase. The chromosomes will start themselves back into the thread-like structure, that is a chromatin. So, these chromosomes, which we have seen that they are going to become the chromatin, they are going to convert themselves back into the thread-like structure, and this is known as a chromatin. But one interesting thing which I want to ask from you is that, students, the parent cell having the 46 number of the chromosomes. Now, how much each daughter cell have? Obviously, that is a 23 number of the chromosome in each daughter cell, because one chromosome, when the chromosomes were in the bivalent, there was a bivalent chromosomes. One chromosome moved to this daughter cell and other moved to this daughter cell. So having the 23 numbers of the chromosome. In the cytokinesis, you can say in the cytokinesis, it is like that there are the contractile rings. These contractile rings, you can say, are made up of actin or microtubule. When these contractile rings are going to get smaller and smaller, this cleavage will become deeper and deeper. And this the point comes that these two cells are going to get separate. So in meiosis 1, a one parent cell basically produces the two daughter cells. It means that a one, you can say, diploid have produced the two haploids. Daughter cell. A diploid means 2n having the 46 number of the chromosomes, while the diploid means 1n having the 23 number of the chromosomes. As well as you know very well about the cytokinesis that occurs in the plant cells, which we have already learned. Now students, after the meiosis 1, the next phase begins, and that is known as the interchinesis, or similar to the mitosis as I told you before, G1S and G2 phase. After that, the meiosis 1 used to begin. The cell used to pass through from G1S and S2 phase. Here, basically, the chromosomes in that phase used to double themselves as I told you before, and having the sister chromatin now. Now, it is going to pass from the M1 like the meiosis 1 phase. In meiosis 1, as I told you before, that they are going to, a one parent cell is going to produce two cells. So, a one diploid cell is going to produce two haploid cells, and these two haploid cells are now going to pass through from the meiosis 2, and they are further going to divide, and now they are going to produce the four cells. That's why it is said that the meiosis is going to produce four cells. After the completion of meiosis 1 and meiosis 2, a one cell is going to divide into the four cells. In meiosis 1, one cell is going to divide into two, and in meiosis 2, two cells are going to divide into four cells. As I told you before that in meiosis 1, the genetic variation used to occur. The thread-like structure is first going to convert into the condensed structure, that is a chromosome, and two centrioles that are present there used to move toward the opposite pole and will start producing the spindle fiber, whereas the nuclear membrane is going to degenerate and the nucleus will also diminish or disappear. These chromosomes are basically going to arrange themselves. The homologous pairs are going to come closer to each other. In the prophase 1, the homologous pairs will come closer to each other and they are going to align themselves with each other, and this condition is known as a synapses. Then they are going to join at some specific point that is known as a chiasmata, and after changing, they are going to exchange their DNA or the genetic material with each other, and this will be known as the crossing over. After the crossing over, each known sister chromatin will have the part of each other. Now, this genetic variation will occur in the prophase 1. Now what will happen? You know it very well. The nuclear membrane or the nucleolus will disappear and the centrosomes will start producing the spindle fiber. These microtubules will be the kinetochore spindle fibers who are going to attach with the kinetochore protein of the chromosome, and each spindle fiber is going to attach each, you can say, a kinetochore protein. Now these bivalent, you can say, tetrad are going to arrange themselves in the center of the cell or the equator of the cell, and that will be known as a metaphase plate. Each spindle fiber will attach from each side, or one spindle fiber is going to attach the one kinetochore protein. Now slowly and gradually, after the attaching of the spindle fiber with the bivalent kinetochore protein, they are going to move toward the center of the cell or the equator of the cell. And when they are going to arrange themselves in the equator of the cell, then that will be known as the metaphase plate. Each spindle fiber is going to attach with the kinetochore protein of the bivalent chromosome. After the arranging of their self in the center, then that will be known as a metaphase plate. Now what will happen? These cells, these spindle fibers, are going to get shorter in the anaphase, and when these spindle fibers are going to get shorter, they are going to pull one chromosome toward each self, and then it will lead to the telophase. The telophase is reverse of the prophase, converting into the thread-like structure of the chromatin. Nucleus membrane will reappear with the side by side the cleavage furrow will occur, and here the cytokinesis will also occur. And this cytokinesis will lead to the separation of the cell, whereas the nucleus membrane will also occur and the nucleolus will also occur. And after the cytokinesis of the of the meiosis1, the next phase, the meiosis2, will start, which we are going to discuss inshallah ta'ala in next lecture. See the video very carefully. If you have any query, you can discuss with me. Allah Hafiz. Assalamu alaikum wa rahmatullahi wa barakatuhu.